Hi, my name is Ryan. I'm here to give you a quick introduction to my program Bandage. Now, if you've ever used a de novo assembler before, then you're probably familiar with the contigs file that the assembler generates. But what's less obvious is that these contigs are actually nodes in a graph that the assembler made, a de Bruijn graph. And this is what the graph file might look like, where you see the nodes and their sequences and the edges connecting the nodes. But this file isn't very easy to work with, and that's why I made Bandage, to visualize assembly graphs in a more convenient way. So this is what an assembly might look like in Bandage. It's a salmonella genome, and what you can see is that there are some regions that assembled very well into long contigs, and some regions that assembled much more poorly into many short contigs, perhaps due to repeated sequences in the genome. So in Bandage, I can zoom in on these areas, I can move nodes around to get a better look at what's going on. I can label them with how long they are or what their coverage is. Or I can give custom labels too. There's different coloring schemes I can use or custom colors. And it's also possible to copy sequences out. If I'm interested in this one, I can copy it to the clipboard and then maybe paste it into BLAST and see what genes are in that region. Speaking of BLAST, I've also integrated BLAST into Bandage, so it's possible to search for sequences within the assembly graph. This is the 16S ribosomal gene, spread over a few different small contigs. Here is the DNA polymerase gene, and that one is nested in a larger contig. So Bandage is freely available. There's a link in the video description, so feel free to try it for yourself. Thanks for watching, and I'll be making more videos in the future to show how to use Bandage in more detail and how it might help you with your assemblies.